So, as I mentioned, today we're going to be talking about a particular structure that is very relevant in biology. The purpose of this talk is meant to be very general and kind of as an introduction not only to this structure but also to resolution. But our focus today is going to be on using the nucleopore complex, which is made up of these proteins called nucleoporins, often referred to as NUCs. So if you've seen the film relevant to the costume today, how do we get resolution that is CNUP? So first, it's important to think about how we define resolution, but why it's also really important. And understanding how finely we're able to see structures in an imaging system actually matters for our choice of instrumentation when it comes to the biological question at hand. So if our system is not able to get us to the resolution of the size of the object that we're interested in studying, it is not going to be a good choice. And this is particularly important in one of the more newer fields of light microscopy called super resolution microscopy. So this is where we're trying to see ultrafine structures that are below what conventional laser scanning microscopy like a confocal microscope can get you to. So that limit is going to be resting around 200 to 250 nanometers. And importantly, as these techniques are trying to work around the diffraction limit of light, like a physical property, it requires some optimization and consideration of the optical components of the system, namely also the stability, imaging conditions, as well as the sample preparation. And so for us, this means that we need to ensure the quality control between systems. So current algorithms might be limited and also require some prior knowledge of the sample in order to enhance those situations. And so we actually, compared to a typical structure that might be fabricated, we need a suitable reference standard that is going to serve for some quantitative optimization of the workflow as well as characterizing the resolution of a microscope. So there's a few methods for defining resolution, but one that's quite relevant to us is going to be based on the Rayleigh criterion. So the resolution of a system describes the ability of a microscope to distinguish fine details or to distinguish between 2 points. So here we're looking at what appears to be two blurred spots together, but if we're not certain exactly what the ground truth is due to the spreading, we need to be able to understand or untangle that. And according to the Rayleigh criterion, what we're going to be doing is defining a system as just resolved if the peak or maximum of one point coincides with the local minimum of the other. And the ability to do this is going to be directly related to the size of the spot that we're scanning with. So the size of the spot that you scan or image with is called an airy disk, and that diameter is directly proportional to the wavelength of light that we use, as well as some properties of the objective, importantly, the numerical aperture. So the wavelength of light, that's the color of light you choose, but the numerical aperture describes this range of angles of acceptance that an objective can have. So the better the objective, you can get a finer resolution, and also the shorter the wavelength, you also get a better resolution too. But another way to measure it and assess it considering this airy disk, again, this equation is very approximate to the one just above it. But we here consider that you can approximate the resolution as the full width at half maximum across these points here too as well. And so how have we previously considered measuring resolution? Well, there's been simulated images as well as using some artificial structures. So this could be anything from a test pattern of line spacing to DNA origami. So this is using DNA, but in such a way as to orchestrate and create these two structures, two fluorophores that can be separated by a very exact concrete amount of space. So here is an example of 90 nanometer DNA origami that can be resolved. But we can also use structures coming from cells themselves, such as histones, mitochondria, microtubules that have very fine, thin filaments, or the beta-2 spectrin periodic helix, which can be precisely measured between 180 or 190 nanometers. So all of these things coming from cells have been visualized across many different modalities, so we know what they should look like. But each of these has some drawbacks. So when we consider using simulated images to model our resolution, there's no real biological basis coming from that information. if we consider artificially made structures, these can be limited in terms of what labels are needed to visualize them, and they might be actually different from in vivo structures themselves as they're changing. So these cell structures, they are a nice candidate, but also the fixation methods can be required, and it can be prone to cherry picking with the current existing structures that have been typically used. So how do we choose something that might be better? So in choosing a structure to quantify a resolution with, it should be labeled and be able to be seen first and foremost with the instrument of choice. If your instrument cannot resolve that structure, it's probably not going to be a good choice. There also should be a defined number of them or have a very exact and precise shape that can be cross-validated across other instrument modalities. This also, too, can lead to the benefit of quantifying labeling efficiency as an extra perk. It should be able to be seen using common dyes and labeling techniques. And for statistics, there should be many copies of that structure. And so here we propose using the nuclear pore complex, which some of you may have heard of. It's becoming quite a buzzword as this very particular structure that has been well characterized and is continuing to be used by emerging super resolution microscopy techniques in particular. So this structure exists across the nucleus on the nuclear envelope, and it represents this porous structure that serves as the sole passage for macromolecules between the nucleus and the cytoplasm. So things are entering in and out here and being exchanged. It has the benefit of the fact that its architecture has been well characterized, especially with electron microscopy as well as other techniques. So here just kind of listing some of the parameters that are shown here as well. But what you can see and hopefully take away is that there are several ranges here. In fact, we have an order of magnitude difference that we could exploit for validating resolution. So down to 100 nanometers for what we would think of as like a superconfocal super resolution system, but anything down to getting into that single nanometric territory. So this one structure in itself is quite useful and can be used across super resolution modalities. And so what is it made of? I mentioned it's this porous structure that's on the nuclear envelope, so surrounding the nucleus itself, where we can exchange molecules, but also it is present where the inner and outer membranes of the nuclear area and the cytoplasm are fusing together. And that nucleopore is made up of nucleoporins. So these are the individual proteins that serve as transport molecules making up the structure. And it has a couple of different layers to it between the cytoplasmic rings as well as nucleoplasmic rings. The two outer rings are going to give support, where the inner ring is going to serve as the transport channel for molecules. And there is about 30 of them, but there's other proteins that play a role in them as well. And they're named typically based on the size of the protein in terms of kilodaltons. But the structure in general is quite interesting for biology because you can imagine, like a blood-brain barrier, understanding and visualizing where things enter our nucleus can affect diseases and create certain pathologies. And in fact, depending on the nucleoporins that may be involved in certain pathologies, they have a different role. So aside from being biologically relevant, there's many copies that are present across a single nucleus. It's a three-dimensional structure, so it can help people quantify their resolution not just laterally, but also axially in depth. It can be stained with common fluorophores and used to also assess, as a little bit hinted to, the labeling efficiency of certain structures. In a technique called expansion microscopy, it can also, because it's so known to be this eightfold symmetric pore, it can be used to classify the expansion homogeneity for expansion microscopy, which swells tissues up to be able to see super resolved structures. It can also be used to calibrate protein counting as well. And so Here, I'm going to show you a few images of what the nucleopore complex can look like across different imaging modalities, but we're going to be considering ranges that span here between a confocal laser scanning microscope and down now to also the single nanometer level of imaging with technologies such as the MinFlux. So here, what we're seeing is resolution near 200 or 250, and we're getting down to 2 orders of magnitude higher. I attended a talk earlier this morning, and the analogy was really great. It's kind of like what you want to see is a single football or soccer ball, if you're coming from the United States, across an entire football field. So here we're only seeing the whole field, and here we can actually get down to finding the individual playing ball. And so how do we get to that place of improving the resolution? When it comes to super resolution microscopy, there's 3 families of techniques that allow us to improve our resolution beyond the diffraction limit. So in one sense, we can try to make the scanning spot size smaller, and this is what stimulated emission depletion microscopy does. So here we're scanning with that confocal airy disk, so spreading in 3 dimensions. But what STED does is then co-aligns a depletion beam. So this donut acts as an eraser for all but a very, very small point that would have otherwise been smaller than what we could scan with diffraction limited techniques. So it optimizes directly the point spread function that the system sees and thus the resolution. We can also try to separate our signal by scanning with something other than a point. This is what structured illumination microscopy does. So by creating either interference patterns or lattices of light that can be shown across an entire image, rotated, we can then collect information, mathematically transform it, and recover the details that in the Fourier space correspond to the high spatial frequency info, which in the image territory corresponds to fine structural details. Or you can change the sample by, instead of having all light on, by emitting or from single molecules at a time. So here, this image of the Eiffel Tower gets built up by taking many pictures of the blinking lights over and over. And this is what Palm and Storm do. So each of these 3 techniques has certain advantages and challenges, but they result in different levels of resolution, and this will be hopefully appreciated on the next slide. So where we begin, here's the example of what the nucleopore complex looks like when you scan with confocal laser scanning microscopy. So here we're using a diffraction limited technique and scanning with an airy spot. So all we can see in that analogy is like the entire football field when we know we should have tiny little points all around here. But it can still help to quantify the resolution of your confocal microscope by seeing how small you can create this radius or these 2 separated points. With techniques that get in towards the super confocal territory, so optimizing and getting to be about almost twice as good as the confocal microscope, the airy scan can then refine the spots to be smaller, and we can also do better. So here with the system Live Codem that uses a raster scanning technology, but instead of scanning with a diffraction limited airy spot, this technology scans with 2 half moons, so some very sharp narrow region of darkness between, allowing one to resolve the inner pore of the inner circle of the nucleopore complex at 107 nanometers in this image. But going further, techniques like STED, where we have the depletion beam, allow us to see even better, higher resolution, getting close to 30 or 20 nanometers, on average around 50, to those more blinking technologies like Palm and Storm. So these stochastic techniques are going to provide the highest resolution of conventional super resolution technologies. But finally, where we're going towards in terms of getting to higher and higher super resolution capacity is with systems like the MinFlux that are going to be able to isolate these individual nucleoporins, and you can see down to single nanometer level. And so this seems like a great opportunity to also mention that there are still some limitations of using the nucleopore complex. So it does require consistent and validated labeling efficiency. which could cause one to risk undercounting nucleopores, especially at the higher and higher resolution techniques. It also means that you want to choose a photostable dye to make repeat measurements to avoid photo bleaching. This system would also still need a good signal to noise ratio, so making sure that illumination is set correctly. If one chooses to use a living sample, which you can image nucleopore complexes using live cells, this could be tricky as there can be variation from cell to cell. Importantly, we need to be able to get the nucleopore complex either flat if you're using a scanning technique in order to see each of the 8-fold points, or resolve the inner or outer diameters. So it's not a structure that's completely immune to cherry picking. But to wrap everything up that we spoke about today very briefly and generally, the nucleopore complex offers a versatile solution to assess microscope system resolution across multiple modalities, in particular for use in super resolution microscopy. It has been well characterized across multiple modalities, so we well know its structure and what to expect. It's biologically relevant, playing a huge role in transport. It's a three-dimensional structure, so can help assess it not only in 2D, but also in 3, and allow one to also characterize other super resolution systems that are pushing ever closer to the nanometric limit, and also useful for optimizing experimental settings, and it can be used, common dyes can be used to image it. And this is all I have for us today. I want to thank you again for your attention, and if you